The interest in single genes have become an important cornerstone in genomic testing. Jason Redding from SA Studbook joins us today to talk about this subject. Why, Jason? Thank you for joining us.
Thank you for having me this morning. That's a really good question. Why are single genes becoming important with the use of genomics in stud breeding? Well, genomics was initially introduced into stud breeding for the use of genomically enhanced breeding values. So you have a reference population of animals that you can then put into the model that enhances the accuracy of these breeding values, especially for young animals. But there are a lot of breeds that don't have a sizable reference population that allows for them to have genomic breeding values. So they don't really see the use or the purpose of genomics, and they've just been carrying on with standard DNA testing for parentage or just for the 3 double muscling mutations. So in order to reframe genomics and get these breeds to adopt genomic technology and build reference populations, we've had to look at immediate benefits, because genomic breeding values is a long-term benefit of genomics. But what is a short-term benefit? I did a previous video on parentage and a few of the double muscling mutations, but there are a wide range of single gene mutations that can have an immediate benefit on selecting breeding animals for these breeders, especially when it comes to the double muscling. There's a lot of detrimental mutations. The pole gene, which is a welfare-based issue. It's favorable if you don't want to dehorn your animals. So it's more of an immediate benefit that the breeders see a value straight away instead of having to wait for genomic breeding values to come into their breed.
Jason, how are recessive genes inherited, and why is it important for us to manage them?
Well, recessive genes are inherited in the same way that any other single gene is inherited. So in my previous video, I spoke about the pole gene, in that the pole gene is dominant over the horn gene, meaning that an animal only has to carry one copy of the pole gene in order to observe the phenotype. Recessive genes work in a similar way, but they act like the horn gene. In order for the animal to be horned, it must carry 2 copies of the horn gene. Recessive genes such as mucopolysaccharides or POMPs or NAGLU, these mutations, in order to observe the phenotype, which is then associated with a disease or with poor calf performance, the animal must carry 2 copies of this gene. And normally these animals are culled. But the breeder forgets that in order for this phenotype to be observed, both the sire and the dam must be a carrier of these mutations. So in order for the mutation to be observed, the sire must pass on 1 mutation and the dam must pass on 1 mutation, but only 25% of the calves become affected, meaning that they carry both copies. But the negativity around this is that 50% of the calves will also be carriers. So if you don't monitor a recessive mutation, it will slowly spread itself throughout the population. They refer to them as the silent killers, because you only see them once the frequency has come to a stage that you are mating carriers with carriers and 25% of the calves are coming out as affected animals.
Well, your colleague, Dr. Helena Tron, actually spoke about the role of the grandfather and the grandmother. And that's what you are referring to, am I correct? Absolutely. Is there an international working group that monitors and standardizes the way mutations are analyzed, reported, and standardized?
So I've just attended the International Society of Animal Genetics conference in South Korea, and there are a few working groups. So there's a working group that most of the labs in South Africa are involved in, where they standardize their testing for DNA parentage. But there's a new group that's come out called the Animal Genetic Testing Standardization Group, or the AGTS group, where we are currently looking at how do we report these mutations to breeders so that they can understand them, as well as are we actually testing the correct mutation? Are we testing for the specific mutation, or are we just testing for a marker that is associated with the disease? And by standardizing the way that we test it, we can do this with Sanger sequencing, but we want to put it on the genomic test. So then instead of having to pay for one, for parentage, or just for pompies, or just for double muscling, if we can put all of these single genes onto the genomic test and package it, which is why we refer to the genomic test as the all-in-one DNA test, then this can allow the breeder to get the best bang for his buck just from a genomic test, because then he gets parentage, single genes, and genomically enhanced breeding values. This working group works closely with the online Mendelian Inheritance of Animals, which is an online website that is going to be used as the standard reporting website of how do we interpret these mutations. Is the mutation validated in other populations, not just the one that it was discovered in? And because I was there and I'm a member of ISAG, I decided to stick my hand up and be a member of this working group for the next 2 years in order to standardize these single genes in a South African context.
Oh, well done, well done. Let's talk about the work you do in the dairy cattle breeds. Which genes of interest have you started to report to the beef breeders in general?
So dairy breeders, especially the Holstein and Jersey societies, they've been selecting for two specific dairy mutations. One is known as DGAT1, which is very well known to increase butterfat production. So we know that the Holstein animals, they have big udders, they produce high voluminous amounts of milk, but the butterfat and protein percentages are quite low. So that's the type of milk that is used for everyday milk use for drinking in your coffee with your cereal. While the Jerseys, they have smaller udders and they produce higher butterfat and higher protein percentages. And this has actually been associated with this DGAT1 gene. The DGAT1 gene increases butterfat production and decreases milk volume production. So there's this negative relationship going on. So the Holstein animals, they don't carry the DGAT mutation. They are identified some animals and they are trying to increase its frequency, while the Jersey animals, known for cheese making, yogurt, and all the other cheese byproducts that we know, they are homozygous or fixed for this DGAT1 mutation. So we started reporting it in the beef cattle, and we see a very interesting relationship with the DGAT, that the European and British breeds like an Angus, Hereford, or a Simmentaler, they are like the Holsteins. They don't carry the DGAT mutation. They have bigger udders, they produce lots of milk. In our indigenous breeds and the Indocene breeds like the Brahman, the Nguni, the Tuli, we see that they are fixed for the DGAT1 mutation. They have smaller udders, but they produce more energy within a liter of milk for their calf. And then what we see in the composite breeds, because they are a cross between Taurine, Indocene, British breeds with Brahman types, most of the composite animals are a heterozygote or a 1. So it's potentially showing an interesting relationship or balance. My colleague Hannah Kruger at SA Studbook has coined the term happy medium, that these composite animals have the mutation, which increases butterfat, but they also carry the wild type, which increases milk volume. So there's a balance in the composite animals that we are seeing. The other mutation we started to report is the A1, A2 milk protein, which is known to affect the digestibility of the protein. The A1 is not as favorable and causes stomach issues in human beings. So if you go to Woolworths or anyone that's selling milk, they've got A2 milk because that's the best digestible milk. And if that is digestible by humans who are mammals, surely it has an effect on the calf's digestibility of its cow's milk. So we've reported that, and there are a few other ones like capsaicin, lactoglobulin and ABCG2, which we don't really see their effect yet in the beef cattle, but we are still monitoring them because as more data comes out, we're going to start getting a better idea of what they do and what breeds that they actually occur in.
Jason, something else we need to talk about is the research on meat tenderness. Quite a few of our cattle breeders associations are focusing on that at the moment. So what is the meat tenderness score that is reported by SA Studbook, and what are the trends you are seeing?
So with the genomic test, I said there are a range of single gene mutations. And on the test, there are mutations on the genes known to assist in the conversion of muscle to meat post-slaughter. These genes are called the culpane and culpostatin genes. These genes switch on when the animal dies and assist in converting the muscle to meat by releasing enzymes that help the muscles degrade a little bit and become tender. There was actually a study in 2022 published by a range of high-standing scientists in South Africa, like Prof. Philip Strydom, Prof. Esti van Marla-Costa, and Prof. Eddie Webb. They're all meat scientists and geneticists. They had 10 different beef breeds in a feedlot scenario. They raised them, slaughtered them, analyzed the carcass data, and then with a genomic test, they associated that these culpane and culpostatin mutations are significantly associated with meat tenderness. So at SA Studbook, in order to provide more value with a genomic test to our breeders, we went and looked at this study, as well as a lot of other studies around the world, because other researchers are looking at these genes in other breeds. And we identified that there is around a 36% additive genetic effect when you add these mutations together. So we standardized that internally, and we decided to give out a meat tenderness score. It ranges from 1, which is normal, all the way to 6, which is the most tender. But remember, this is just the animal's genetic ability to have tender meat. There are other factors such as the environment in which they were raised in, the slaughtering process, as well as the chilling of the carcass. And at the end of the day, how does the person cook the meat in order for it to be tender? So it's just another way of looking at how can we give the best product to the consumer at the end of the day. What's interesting is that the breeds that are known to be associated with meat tenderness, like the Senepals, the Brown V's, the Angus, most of their animals are a level 4 or a level 5, meaning that they carry these mutations for the culpane and culpostatin. But you will see from the figure that I have provided in the article in this month's stock farm that 50% of the animals that we have tested in South Africa are in level 3. So it's nothing to be worried about. Most animals are a level 3, but if you are a breeder or a commercial farmer that's looking at improving your end product, it could be good to start selecting for these animals that are a level 4, 5, and 6 to increase the mutations that we are seeing that have an effect on meat tenderness post-slaughter, especially on 3 days. There's no correlation with the 30-day because the aging of the meat is the biggest effect on tenderness. But most of the meat that we eat in South Africa that you see on the shelf is between 3 and 10 days. So it potentially will have an effect on the consumer's preference or consumer perception of the meat product at the end of the day.
Very informative, Jason. Thank you very much for joining us. Jason Redding from SA Studbook, talking to us about the interest in single genes and how they have become an important cornerstone in genomic testing.